I'd like to introduce our speaker, Dr. Tim Hefferon. He is originally from upstate New York and received his undergraduate degree in biology from the University of Virginia, and then he went on to get his PhD in human genetics from Johns Hopkins University. Since then, Tim has been working at the National Human Genome Research Institute in the lab of Eric Green. His main interest is in comparative genomics. That is, he compares the genomes of other species to ours to try to learn more about the human genome. And he's also interested in bioethics. Tim's work in PXE centers around finding difficult to find mutations, ones we can't identify very easily. And with that, I'll hand it over to Tim. Welcome, Tim, and thank you for being here.
Thank you very much, Chris. Good evening, everybody, and thank you for coming. This session should teach you a little bit about the basics of genetics and of PXE and the ABCC6 gene in particular. I do encourage a lot of questions. That's the best way for us to work this. So please heed Chris's advice and type your questions into the question pane, and I will try to pause every so often to make sure that I'm addressing any pressing questions that people have. So to begin, you all know that the human body is made up of cells, and a whole lot of them. The average person has about 100 trillion cells. That is 1 with 14 zeros after it, which is more than I've ever counted. These are all different kinds of cells, bone, skin, nerves, muscle, etc. But the one thing that all of these cell types have in common is that every single cell contains 2 copies of the human genome. One of those copies you inherited from your mom, and the other copy, of course, you inherited from your dad. And both of these copies, as I say, are present in each one of those 100 trillion cells in your body, with a sort of disclaimer here, which is to say that red blood cells used to have 2 copies of the human genome, but as they mature and develop into the cells that they where they can serve their function in your blood, they actually lose that information. So with that exception, every cell has all of this tremendous amount of information encoded in the DNA in the cell nucleus. There are about 20,000 genes in the human genome. You have, of course, if you have 2 copies of the human genome in each cell, you have 2 copies of each of those 20,000 genes. 20,000 that you got from your mom and 20,000 from your dad. I should also say that the genome, the copies of the genome in each one of those cells in your body is identical from one cell to another. The only differences, of course, are the version that you got from your mom and the version that you got from your dad. And those versions are just copied into every single one of your cells. So this will compose mainly three parts in this webinar. One is discussing what DNA is, also what a gene is, and finally what a mutation is. And after I have gone through these and hopefully answered your questions, we will also have a small section about genetic testing and what you can and cannot learn from that, and when's a good time to have that testing if you choose to have it. But to begin with, we'll talk about what DNA is. DNA is found in all living things. It's the instructions, essentially, for building the body of whatever thing the DNA finds itself in. So if you actually take a sample, say a blood sample from a person, as you may have done, and you go through the process in the lab of extracting, taking that DNA out of the blood and purifying it, so that you can study it, it looks like this fibrous white stuff that you see in this test tube in the center of the slide here. And that's going to look the same. The stuff itself is going to look the same whether it comes from an animal or a plant or a person or a bacteria. The DNA encodes information, obviously, and that information is different depending on what organism it is found in. And a couple of the neat things about DNA is that it stays essentially the same throughout the lifetime of the person, and it copies itself very very faithfully. So it makes exact copies of itself every time a cell divides, and that's going on in you right now, as well as you developed from an egg, from a fertilized egg into an adult. The information is encoded in the DNA double helix. You've probably heard about that. It's essentially a long string of letters that gets coiled up into things called chromosomes, and these chromosomes are found in the nucleus of the cell, every one of the cells that we talked about, found in your body. Just a couple of DNA facts. I already said there are 20,000 different human genes. Each of these genes contains the information to make a protein. And proteins are the sort of the machinery and the stuff that makes up the structure of your body, and they do the enzymatic reactions, the chemicals, hormones. All of this structural stuff is made out of proteins, as opposed to the DNA, which just sits inside the nucleus and basically doesn't do anything except everything. It contains all of the information to make these proteins, and the proteins then go and do things. DNA is tiny. It's so small you could fit about 5 million strands through the eye of the needle. So obviously hard to see without a microscope. And if you took the DNA in just one of your single cells, one of those 100 trillion cells, it would stretch out to about 6 feet if you pulled it end to end. So given how tiny it actually is, that's a lot of information contained in the DNA. So DNA consists of 4 building blocks. It's just a long string of letters, one after the other, and it's at each point along that line, there's either an A, a T, a C, or a G. And that's all DNA is, essentially. It's a long string of these building blocks of A, C, T, and G. And it's the order in which these letters are found that winds up being very, very important. Obviously, it tells the difference between making a bird and making a mouse, for example. So here's just an example of a long stretch. You can see this long stretch of A's, C's, T's, and G's. And it's really hard to imagine that this seems to be really simple, but it contains a whole lot of information, not only in the order of these things, but in the way that they're put together into sort of larger words and sentences that we can't tell from just looking at this sequence like this. So this is just an example of something that you've seen, DNA sequence. Okay, before I go on to ask what is a gene, we'll ask if there have been any questions so far on anything. If everybody can hear me, this is a good time to check to see if everybody can hear okay and everything.
I don't have any technical issues that have been reported, so I think everyone can hear. And there's a question that I think will go better later on, and it's probably about, what will the children inherit? So.
Okay. I'll try to spend a good amount of time on that. Okay, moving on. What then is a gene? Well, a gene is simply a term for a section of that DNA, a stretch of those A's, C's, and T's, and G's that we talked about. There are 20,000 genes. As I said before, each one of them contains information for making a protein. And I think, as you can appreciate in this slide right here, the proteins that are made from different genes, different stretches of DNA, have very different appearances and very different functions. They're built to do different things in the body, in the cell and in the body. So for example, the one over on the left from gene 1 encodes a protein called lactase. This is an enzyme that breaks down the sugars that are found in milk. And obviously, if you know anyone who's lactose intolerant, one of the reasons they can be lactose intolerant is if they have a defective copy of this gene or a version of this gene that doesn't break down those sugars as efficiently as it could. Likewise, all of the other proteins here serve different functions in the body. And if the sequence of the DNA from a given person's copy of a gene is different, then the structure might be a little bit different, and you can get variation in, in everything from height, person's height, to skin color, to how, again, how they break down lactose, to really anything. These differences in the genes are what are really responsible for the differences, all of the differences that we see between people. The other thing that's different about these genes, I said that the information in the genes is identical in all of your cells, but the genes can be turned on and turned off also by the cells. Obviously, a nerve cell is going to need different proteins and different functions than, say, a muscle cell is going to need. And so there are ways that the cell's machinery can turn on the genes that are needed and turn off the genes that are not needed. So for example, you have these different cell types, and if you look over on the right, those are your identical copies of the genes, but different genes are turned on or turned off depending on what type of cell they're found in. This is why all of your cells don't look the same. They're different. In addition to the genes, one may wonder, how are these genes turned on and off? Well, one of the things that we've been finding through some of the comparative genomics that Chris mentioned that I study in the beginning is that in between genes there are these regions that are colored green here, and those are what are called regulatory regions. They don't actually code for any protein, but they do have the information that tells the cell what genes need to be turned on and off and when. So that these regions that, before we understood them, we still don't understand them very well, but we've heard the term junk DNA. If you actually look at all of the genes, there's the yellow parts here in the genome. The yellow part only accounts for 1.5% of the entire genome, and the other 98.5% used to be thought to be junk DNA. Well, now we know that actually it contains a lot of these signals that are a critical part of how the genes work by turning them on and off. In terms of, again, the genome, it's I said there's a lot of letters. There's 3 billion letters in each copy. So if you printed these out in terms of a good thick 1,000-page phone book, with one copy of that genome would fill about 200 of these phone books. And remember, inside of every single cell, you have 2 copies. So you would have 400,000-page phone books. You do have the equivalent of 400,000-page phone books coded for within the nucleus of every single one of those 100 trillion cells. So it's just, it's hard to think about how much information this is and how small it must be. But there you have it. So each person does have a unique DNA sequence, but it's only different in a very small fraction of the bases that are there. So here you have on the left, you have 2 different people, and if you take a sample of a cell from each of their bodies and you look at the DNA sequence, most of it is exactly the same, over 99% of it. In fact, a lot more than 99%, something like 99.8%. Because you've probably heard that we share 98.5% of our DNA sequence with chimpanzees. So most of the DNA is exactly the same, but it does vary in these little points that are highlighted in yellow here. One person may have a G, while one other person at that equivalent position has an A. And it's the cumulative effect of all of these little differences in our DNA that again makes us different and unique from one another. Just, let's see, yeah, here we go, 99.9% identical. These differences happen about 1 every 1,000 base pairs on average. and that if you add all of the differences up, these little differences, there's about 3 million of them that differ between one person and another. And of course, as I said, if the DNA sequence is different, then you may get two different versions of a protein. You can see here that because these DNA sequences are different, the proteins that they produce may look a little bit different and function a little bit different in the cell, and that results in a difference in how healthy somebody can be, or what they look like, or what their muscle structure is, and so forth. And of course, if you have two people who have exactly the same DNA, these are identical twins, and that's because they have all of the same information, and their cells, the genes, turn on and off at the same time, and basically this just goes to show you how important DNA is in making a person. However, DNA doesn't act in a vacuum. You know that what you eat and your lifestyle choices, what sort of chemicals you may be exposed to, they can certainly all impact your health. And in fact, there is an interaction happens between your environment and the things that you do and the things that you're exposed to, the way that that interacts with the DNA and your genes that results in the final picture. They call it gene-environment interaction. Both environment and your genes are very important in your overall health. So to speak about PXE in particular, one of these genes of your 20,000 genes, as you can see here, is called the ABCC6 gene. That encodes a protein. The protein, we don't know what it does exactly. We do know that it transports something across cell membranes. Recent research has suggested that it's involved in the transport of a form of vitamin K, but research is ongoing to figure out exactly what this thing does in a normal situation in the human body. This is the DNA sequence of the ABCC6 gene. I should say, if I haven't said already, that the ABCC6 gene is the gene that has mutations in it when a person has PXE. This is sort of the expected DNA sequence, comprising 4,500 letters of A's, C's, G's, and T's. And now I will go into what a mutation is. But first, again, I will pause to ask Chris if there have been any questions so far that are appropriate at this time.
Yes. Someone asked, can you explain what MRP6 is in relation to ABCC6?
Sure. MRP6 is just another name for ABCC6. It's the same exact thing. It's just, research works that way. There wind up being a lot of different names for the same genes, the same proteins. So MRP6 and ABCC6 are the same thing.
Thank you. That's all the questions for now. Mm-hmm.
Okay. So what is a mutation? Well, if you have the DNA sequence of the ABCC6 gene here, one of the most common mutations that is found in patients with PXE is called R1141X. And the only difference between the regular DNA sequence and the R1141X mutation is that the C changes to, actually, what does it change to? I should know. I believe it changes to an A. I thought that was in my slide. I apologize. But it's just one little letter that has changed. And that seems like something very small, but in fact, as the cell reads through this DNA and starts making that protein that we talked about, this change that happens is in fact what's called a stop signal. And so the cell is making the protein, it gets here and it says, oh, this is telling me I should stop. And so it doesn't make all of the rest of the protein. So obviously that little change has a big effect So that's one kind of mutation. Another common mutation that is found in PXE patients is when this whole section of DNA right here is deleted. It's gone. So the cell comes along, it's making the protein, and it just skips from here down to here and keeps making the protein. But again, obviously this is going to have a huge effect. If all of this information is missing, then the protein that comes out of this is going to be very different than what the cell expects. In addition to those two common mutations, there are more than 250 others, some of which are very rare, but they all involve some change in this DNA sequence here that you see before you. So I know that can be a little bit new for some people, so I'll ask, are there any questions about that?
Someone has asked a question about are there other genes that contribute to causing PXE? Do we know of any others?
There are frequently what are called modifier genes for some genetic diseases, which can alter the way that the clinical characteristics of a disease. But the first thing that you have to have, and that you do have if you have PXE, is mutations in the ABCC6 gene. Now, in the context of all the other genes in your body and environment, like we said, yes, that can have an effect on things. But the fundamental molecular cause of PXE is always going to be two mutations in this DNA sequence.
Okay, and then another person asked, all of those 250 mutations that we know of, do we know they all cause PXE?
That's a good question. We don't. All that we know is that they are DNA sequences that differ from this. This is sort of what we expect in people who don't have PXE. And so if you take a person who has PXE and you examine their DNA, you find a difference. To some extent, you assume that that's the cause of their PXE. But that is by no means proven. If you were to sequence as many normal people, you might also find some changes in here because you never know with a given change if it's harmful or not. But generally, when we say that we have these mutations, we are saying that we found these DNA differences in patients and that we didn't find them in normal individuals. So there's a good chance that these mutations are in fact disease-causing, but there's no proof.
And a corollary question is, how certain are we that ABCC6 is the gene? That
100%.
Okay, good.
We do know that, yes, we do know that that is the gene. And we know that through various statistical things, but also because every PXE patient that you find is so far, I will qualify this in a second, but they all have these, like I said, the R1141X mutation, the del23 to 29 mutation. These are mutations that you don't find in people who don't have PXE. And I was going to say, the qualifier was that there are PXE patients where we have not been able to find mutations in their genes. So either we have missed them for some reason or another, or there's something that we don't understand. They may have a mutation in one of those regulatory regions that's not in this sequence, for example, that tells the cell when to turn and turn off the ABCC6 gene. These are some of the difficult-to-find mutations, again, that Chris mentioned that I was working on at the beginning of the webinar.
Okay, thank you.
Okay. So, to talk a little bit about genetic disease in particular, generally speaking, it can be divided into two kinds. One is called dominant and one is recessive. Dominant genetic disease requires only one mutation It can be in the copy that you inherited from your mom or in the copy that you inherited from your dad. It doesn't matter. If you have a mutation, you have a disease. And what this means is for a parent who has that disease and has that mutation, 50% of the time when they have children, they're going to pass on that mutation to their kids. And so in the case, again, of dominant disease, if you have a mutation, you have the disease, that means that 50% of their kids will have the disease. And for their kids' kids, again, if a kid who has inherited the disease from the parent, 50% of the time they will pass it on to their children. So these are the characteristics of a dominant disease. Now, I want you to forget all of that because it doesn't have anything to do with PXE. Fortunately, PXE is a recessive disease, which means that you have to have two mutations, one in each copy of the gene. So in order to have the disease, PXE, you in fact have to have a mutation in the copy that came from your mom, and you have to have a mutation in the copy that came from your dad. And also what that means for parents who are both carriers, I should say a carrier is a person who only has one copy. So a carrier does not have PXE. But the way that that people do wind up with PXE is when two people get together, each of whom have one mutation, and when they have children, 1 out of 4 of their kids is going to have PXE. And we'll go into some more detail about how that happens, and I can certainly answer questions about specific cases of carriers and people who have disease. But also, more good news is that there's a much lower chance of passing the disease on to the grandchildren, because not only does the child have to be the 1 in 4 who has the disease, but they have to wind up marrying somebody who also has a mutation in the ABCC6 gene. And generally speaking, this is a very rare disease. So the chances that someone that you meet out there is going to have a mutation in the ABCC6 gene is really low. So the chance of passing it on to grandchildren is generally very, very rare. So keep in mind as we go through the following slides that PXE is in fact a recessive disease. Requires 2 copies, 1 from each parent. I'll stop to ask if there are any questions about that.
So maybe the question about a person with PXE, can you describe what we know about their children?
Okay, we know, that's a good question. We know that a person with PXE has 2 mutations. So every child that they have is going to get one of those 2 mutations. But getting 1 mutation doesn't have a detrimental effect at all. Their children from just the 1 mutation will just be a carrier, and they will be completely healthy and not know anything about PXE. In the rare case where that child who has 1 mutation, in the rare case where they marry somebody who also has a PXE mutation, then their kids would also have a 1 in 4 chance. 25% of their children essentially would have disease. But we do know that the children, all of the children of someone with PXE are carriers.
And if the other parent is a carrier, and so they wouldn't know that they are a carrier because they wouldn't have disease, what are the chances that the other person is a carrier? So how frequently is the carrier status found in the population?
Oh, this I should know by heart. I believe I would have to. I would actually have to look that up. I don't want to give incorrect information.
So that's the other concern for a parent for parents with PXE is what the spouse, what their status is.
That's correct.
Yeah. Okay. That's all for now.
Okay. And I don't know if is Sharon Terry on the line?
She is not, and neither is Lionel.
Okay. They would have that information.
They would, yeah.
About the frequency, and I can certainly retrieve that before we close tonight.
Okay.
Okay. So if you have a mom and a dad, each of whom have 2 copies of the ABCC6 gene, let's walk through a scenario so we can understand a little bit more about how genes get passed on and what chances are of children having PXE and so forth. Let's assume, for the purposes of this, that mom has a mutation in one of her copies, dad has a mutation in one of his copies. So these 2 individuals do not have PXE, but they are carriers. So let's go through what could happen. One possibility is that mom could pass on her normal copy, and dad could pass on his normal copy. That's going to happen one out of 4 times. And the result is somebody who doesn't have any PXE mutations. Another thing that could happen, another 25% of the time, is that mom could pass on her mutation and dad not, and that results in a carrier. Another possibility is that mom passes on her normal copy and dad passes on his mutated copy, in which case, again, you have a person with 1 mutation who is simply a carrier. And finally, 25% of the time, mom will pass on her mutation, and that will meet with dad also passing on his mutation, in which case you would get an individual with PXE. So if you have PXE, then this is one possibility, is that your mom is a carrier and your dad is a carrier. So let me put that another way. If you have PXE and your parents do not, then this is exactly what happened. Mom had a mutation, dad had a mutation. You had a 25% chance of getting both mutations, and that's what happened. The picture is slightly different if you have PXE and one of your parents has PXE. And if anybody wants to go into that, we can go into that later. But just for now, for purposes of understanding this, mom and dad are carriers. 3 out of 4 children will not have PXE. 1 out of 4 children will have PXE. And this could be, we're going to get into a little bit about genetic testing, and this could be one thing. And I think the question just asked sort of alluded to this. You know, if you have PXE and you want to know if the person that you're planning on having children with carries a mutation, that may be one reason that you would want to get genetic testing done, so you could know what the chances were of you having children with PXE. Okay, any more questions before we go into testing?
No, not right now.
Okay. What is a genetic test for PXE? This is simply a lab test that you get from either a cheek swab or a blood sample. We study your DNA, your ABCC6 gene, and look at that DNA sequence to find out whether or not you have mutations in that gene that are associated with PXE. Why would you consider the test? Well, theoretically, if you wanted to intervene medically so that you could have, go to a doctor more often, have them look, knowing that you have a mutation, they might look more carefully for signs of PXE. You could avoid risk factors that are known to be harmful to PXE patients. And you could make lifestyle changes to address these risks that you might be faced with. And finally, another reason is to relieve anxiety. If you have a test and it's negative, then that's a good thing, keeping in mind, of course, that we are never 100% sure. As I say, we have patients where we have looked at the DNA sequence for mutations and not found any, and yet it's obvious that they have PXE. So it's not a 100% foolproof thing, but it could certainly relieve your anxiety if you're worried about it. Some things that you might want to consider before being tested is that you could have emotional impact not just the release if they don't find any mutations, but certainly if you do, is that something that you really want to know, especially if there's not anything substantial that you can do about it. You might want to think about that before having a genetic test. Likewise, it could cause family tension. There have been cases, not with PXE that I know of in particular, but with other genetic diseases, such as Huntington's, for example, where when one person finds out whether or not they have the mutation, that has implications for other members of the family who may not want to know anything. So there can be some tension in the family that arises from sort of finding out this information. Again, it could provide you a false sense of security if mutations are not found. As I say, some patients we don't find the mutations, so it's not 100%. Whoops, excuse me. There could be unclear results, meaning, again, the test is not 100% foolproof. So you may say, oh, well, we found a DNA change in your gene, but we've never seen it before in a patient or anybody else, and so we don't really know if it's harmful or not. Genetic tests are certainly expensive. Even, I think PXE International has done a lot on trying to get insurance companies to cover it. I'm not an expert on insurance coverage. Again, those questions can be addressed on the PXE website, and I'm sure that Sharon could address them.
I could tell you a little bit about the PXE test, and this is a question that came up. It does cost, the level 1, tier 1 costs $500 approximately, but if they find no mutations in that level 1 and go all the way up to the top level, it can cost up to $3,000.
Right. I think that's on a future slide.
Oh, okay.
I'm not sure.
Sorry.
But yeah, that's exactly correct. And then, as with any genetic disease, there are confidentiality concerns. If there have been incidents of insurance companies denying claims to people who are denying insurance rather to people who may have genetic mutations. So it's this issue of privacy. You don't want your genetic information to get out into the wrong hands. Employers or insurers who might discriminate against someone with a certain genetic profile. What do the results mean? If you do have 2 changed copies of the gene, then you are affected by PXE. If you have 1 changed copy of the gene, then either you are a carrier or you are affected, but the test wasn't able to detect the second mutation. So either of those is a possibility if you come back with 1 mutation. If you have no mutations, then it's likely that you don't have PXE, because as unusual as it is to not find to find 1 mutation but not the other, it's a whole lot more rare to have PXE and to not be able to find either of the mutations. That is an extremely rare circumstance. Limitations of genetic testing for PXE is that not all the mutations have been discovered. Again, if they find a new variant in your DNA that has not been seen in anybody else, they don't know whether it's harmful or not. The way that the testing works is it first looks for the common mutations, and if those are not found, then a more rigorous, more expensive test is undertaken to see if other mutations can be found. Again, these are some of the more common mutations. And let's see. So as we say, not all of the mutations are known, and we haven't determined if or how these different mutations affect the clinical outcome. This is one of the things that we try to learn when we study genetic disease is can we say, oh, if you have this particular mutation, then you probably will have more mild eye disease, for example, in the case of PXE. Research has not borne that out. It doesn't, in the case of PXE, there does not seem to be any association between a particular mutation and a particular aspect of the disease. And this is one reason that you might not want to get genetic testing is, what is it going to tell you? You don't, all you know is, if you have PXE, you know that you have 2 mutations, you just don't know what they are. And when you have genetic testing done, well, now you know what they are, but you still can't do anything about it. So that's one argument against it. However, there are circumstances in which it's a beneficial thing to do. Like we talked about, if you're contemplating having children with someone and you want to know, you have PXE, you want to know, are they a carrier? That's certainly one example where you would want to know, you might want to know. But in the absence of, a clear path forward, if you have a certain mutation, if there's no clear path forward, then you might want to question why you would want to have genetic testing done. I should say, having said all of that, there are certain kinds of mutations for which there's potentially promising treatment, not for PXE specifically, but in general, and that's these mutations that I talked about, like that R1141X, that send a stop signal to the cell. There are drugs that can be given that are showing some promise that are able to hide that stop signal and basically say to the protein, well, don't look here, just keep on reading. And the cell will keep on making the rest of the protein. These are some drugs that are under clinical trials and things that are hopeful for certain kinds of mutations, those kinds of mutations that produce a stop signal, for example. Okay, how much does the test cost? If you are a new patient and you go in and you ask for a genetic test for the most common mutations, that is $500, as Chris mentioned, and it takes about 2 to 4 weeks to get results. If your mutations are not found and you want to look in more detail and deeper into your gene, you can look across the entire gene for rare mutations, and that costs $3,000 and takes 8 to 10 weeks. It's much more involved. If you already know your mutations and you want to know whether a relative in your family has that same mutation, then that's a quicker test. You can do that for $350. Testing of a relative for 2 specific known mutations, $500, again 2 to 4 weeks. And prenatal diagnosis can be done for a specific known mutation for $1,500, and that also takes about 2 weeks. So before we go to the last slide, I'll ask if there are any questions about testing or anything else.
Yes, we have quite a few that came in.
Okay.
First, about the test, do you know which of the common mutations are tested for in the first tier? How many of those 250 known ones?
I don't know the details, but this is Sharon's slide here showing the common mutations. So certainly at least these 9 mutations are tested for in the first tier.
Mm-hmm.
I think through the PXE website you can probably learn a lot more of the details about how many and which mutations are tested for.
Yes, I think the first tier does cover the mutations that cause about 75% of the known cases, but I'm not sure which mutations those are. But it's a small number compared to the 250.
Right.
And then could you talk again, I know you did address this, but does the mutation, does a particular mutation have any relationship to the physical signs of PXE that a person has? Do we know any?
Nope. This has been looked at again and again by PXE researchers, and they have not found any association between a particular mutation and a particular aspect of the disease.
So knowing your mutation wouldn't give you, just to know the name of it, wouldn't give you any special value that you could, any information that you could use to alter your course or anything like that, or even know what kind of manifestations you'd have? Not
at this point.
Okay.
No. If you're like me, you probably would just have to know just for the sake of knowing. If I had, $500 lying around that I didn't know what to do with, then I might give in to having to know. But it won't make any difference in your treatment at this point.
Right. Okay. And then there's some confusion about the chances of PXE passing PXE on to your children if the two carrier scenario. So I think you said there was a 1 out of 4 chance that your child would have PXE if the two parents are carriers.
Right.
I guess the confusion is, does that mean if you have 4 children, only one of them will have PXE?
No, it does not mean that. This is all statistics. If you have 2 carriers, you know, in some cases, and in fact, at the PXE meeting in Bethesda back in September, I met someone who where both parents were carriers, and something like half of the children had PXE. You know, it's a flip of the coin. It's the same way if you flip a coin 5 times or 10 times, you may get 5 heads and you may get 5 tails, or you could get 8 heads and 2 tails. It's all in the numbers.
So it's each conception has that 1 in 4
right.
chance.
Exactly. Every single conception has a 1 in 4 chance.
And it's a Terry family. They have 2 children, and they both have PXE. So their flip of the coin was not the lucky one either. Exactly. Or maybe it was lucky. I think that's all our questions for now. Nope, no, wait a minute. I think it's another question about the mutations, but I'm sorry, I'm not understanding the question. This is from a gentleman in Massachusetts, and so I will try to get his question answered after. Pardon?
Or perhaps he could ask it over audio?
Not at the moment. He can't.
Okay.
So, well, maybe I could. Let me see. Let me see if that will work. That would be interesting. So I have to find him. Okay. Robert Chase, would you like to ask your question out loud? I bet he is.
I'm not able to hear anything.
Yeah, I can't hear anything either, and I bet he is probably just listening on his computer and isn't connected to a telephone. So there are no new questions coming in, and thank you for this very informative presentation. If anyone has any questions they think of later, they can send them to me, and I will answer them myself or ask Dr. Hefferon to answer them. Whoop, I have one more question. This is again about the chances of a grandchild of a person with PXE becoming a carrier.
Okay.
What are those chances? Can you show that? Can you go back to that diagram?
Sure. So the grandchild of a patient with PXE?
Yes.
Okay. So all of the children of a patient with PXE are going to be carriers. That means they're going to look, well, I should backtrack on that. All of the children of a person with PXE will at least be carriers. Most of the children, in most cases, they will just look like this. They will have one mutation. If this person marries somebody else who also has a mutation, then that changes, because if that mutation gets passed on and combines with the mutation from the person with PXE, then you'll get a PXE child. But let's see. So if we want to go back to all of the children of someone with PXE will at least be carriers, then in order for any of the grandchildren to then have PXE, this child would have to marry somebody else who either had PXE or was a carrier to have any chance of having grandchildren with PXE.
And the chance of having grandchild as a carrier is for if the carrier has children.
If the carrier has children, then 50% of their children will be carriers.
Or 50% chance each pregnancy.
Exactly, 50% chance each pregnancy that the child will be a carrier.
So it gets less common.
Yeah, again, you know, a carrier is not harmful at all. The only implication of someone who's a carrier is that if they marry somebody who either has PXE or is a carrier, then there's a chance that some of their children are going to have PXE.
Mm-hmm. Okay. Thank you. There's a question about what the gene is actually doing, the mutations are doing. Does it look like this is a condition where the faulty protein is, let's see, is it a missing protein that should be circulating in the bloodstream is what he's asking, based on what we know.
The protein itself that is created by the ABCC6 gene is found in cell membranes, mostly in the liver and in the kidney. That's the main place that it's turned on, and it stays there. The problem is that it's supposed to transport something across those membranes, some chemical, and if it isn't able to do that, then there's an imbalance of that chemical, whatever it is, in the blood. And that is sort of the model that we expect is the way the PXE works, is that whatever this this chemical is that is supposed to be transported by ABCC6 can't be transported properly and circulate throughout the body and has the effects that you see in someone with PXE.
Mm-hmm. And presumably that whatever that is prevents mineralization all over the body.
Exactly. If it's normal, if things are operating normally, then you don't get mineralization. But as soon as you disturb that that chemical, then you get this mineralization that causes problems.
Okay, we have thank you for that. We have another question about potential gene therapy to prevent PXE when the egg is a zygote. Is there anything on the horizon in gene therapy?
I don't believe that there is. I'm not familiar with gene therapy attempts in PXE. I will say that in general, gene therapy trials for all genetic diseases have been mostly disappointing. You know, everybody's very eager because it sounds like a magic bullet, but it's been very difficult to accomplish. And as I say, I'm not aware of gene therapy for PXE in particular. And you all are probably more familiar than I am with the various treatment options that are available.
There aren't any trials of any treatment options currently taking place. Hopefully, there's one planned for treatment of the eye, but that's using current drugs of astenomycin, and it's not based on gene therapy. There's no gene therapy trials at all for PXE.
Okay.
And you have answered this, but it can be very confusing. Can genetic testing tell you if you are a carrier?
Genetic testing can most likely tell you if you're a carrier, meaning that we look at the DNA and have a very high likelihood of finding a mutation if you have one. It's not 100% foolproof, I have to say that.
Right.
But most likely, if you have a mutation, we will find it, or the testers will find it.
And because the results can be confusing, PXE International offers to anyone who wants to have the test, we offer genetic counseling at our expense. So we would like you to be able to understand your results, and they can be confusing. You could have a result that said they found one mutation, but you clearly have PXE. So how does that, how do you take those two bits of information, your clinical diagnosis of PXE and the fact that they only found one mutation? So if you're interested in the test, contact us about genetic counseling. And genetic counselors are very good at helping you understand genetic test results.
They're outstanding, and I can't stress how important that is, that if you do choose to have a genetic test, develop a good relationship with a genetic counselor, because they're amazing.
Yep.
And very helpful.
Tim, we have a question about whether the protein that is not circulating in the cells, circulating out of the cell, might be replaced by injections. And I don't know that that's necessarily your area of expertise, but would you like to take a?
No, it's not, and so I don't know the answer to that. And I would just say that they're still working on finding out exactly what that chemical is that is supposed to be transported, and how the disturbance that happens causes the symptoms. So I'm not able to be much help there.
Tim is participating, and a lot of other researchers are participating in a meeting of researchers on PXE, which will take place just right after Thanksgiving, and hopefully we'll all know a lot more about what everyone is researching and thinking of that substance might be. There's a lot of candidates for what might be circulating or missing in the circulation of people with PXE, so we wouldn't even know what to try to inject at this point because we don't know what that is. I think that's it. And Tim, thank you very much for being with us tonight. We really appreciate you taking the time to educate us on the genetics. They're always a little confusing.
My pleasure, and Chris, thank you very much. You've been very helpful.
You're welcome. Thank you.